University is going to give a talk.
Any more of those?
Brief? Ellen's going to be talking about a lake trout project she has going on, where they've been embedding transmitters in these fish and tracking them with receivers in different parts of the lake. So, good stuff. Thanks for coming.
Thank you for the invitation, and I know the afternoon is already quite late and you've been very patient, so I'm going to give you a short version of what I could go on for hours about, but I'll take about 10 or 12 minutes over this. And basically what it is, can we dim light just a little bit? Yep. If that's possible. Back here or back down there? Basically, what I want to introduce you to is a new technology that we've been bringing into the lake, have brought into the Lake Champlain Basin that we're hoping will inform a number of different studies. I'm interested mostly in lake trout, but we're already expanding it to walleye, and the state is interested in expanding it to sturgeon. And as Bill mentioned, very simply what this is a remote method of tracking fish in very short intervals of time for long intervals of time. I'll explain what I mean by that. This is acoustic telemetry, and the idea is that you surgically implant a tag inside of a fish. That fish will emit a uniquely encoded signal, so you know which fish you're hearing, and that signal will be transmitted usually on average between 60 seconds and 180 seconds. So on average about 2.5 to 3 minutes for, depending on the type of tag and the size of fish you've got, up to 3 to 10 years. So you tag the fish once, and imagine tagging a lake trout on its spawning ground, for example, then you'll hear it again at that spawning ground if it comes back the following year, and then you'll hear it again the following year. Or if you tag something like a sturgeon, you can listen to it for 10 years of its life. Given that a sturgeon lives for up to 40 or 50 years, that's a significant portion of that sturgeon's life. So what we've been doing, I've got a graduate student, Tori Pinheiro. She has learned and has taught me now how to do the surgical implantation of these tags. This is, as you all know, a lake trout belly. That is a lake trout, right? Just checking it wasn't one of our walleye. And that's the tag. It's this small, like, bullet-shaped thing that goes inside the fish. We suture the fish. We put them into a holding cage. They recover. They're great. You know, after the anesthetic, they recover very nicely. Let them go 12 to 24 hours later, and off they disappear. And then the other part of it is that we have to put receivers in the lake to pick up those signals that are being transmitted. So the receiver you're seeing here on the right is that black, again, bullet-shaped, but now much larger device that is put underwater, and it's on a submerged buoy system. So for most of the year, these things are not visible from the surface. They're way down below the water. We buoy them at intervals so that we can pick them up with a smaller boat, download the information off the receiver, and then put it back in again. So the batteries need to be changed out about once every year and a half, but the information from them needs to be downloaded every 2 to 3 times a year. We currently have 27 of them in Lake Champlain. Now, to give you a perspective on what this technology is all about and where we're getting the inspiration, this is the Great Lakes system. It's known as GLATOS, the Great Lakes Acoustic Telemetry Observation System. They have over 600 receivers out in the Great Lakes, and they move around from year to year. They are used for different studies, but they're studying the movement of, like us, walleye, sturgeon, lake trout, sea lamprey, and a few other species. In total, about a dozen species are being studied this way. But as you can see, they've got one of the biggest arrays of these receivers anywhere in the world. These things are also being used in Europe, they're being used in the Atlantic, there's some in the Pacific, there's some in major rivers. But the Great Lakes have got them basically throughout the lakes except for Superior. And so we took our inspiration from them. Of course, we called ourselves CATOS, in honor of UVM, the big cats, right? But Champlain Acoustic Telemetry Observation System. And this is where our receivers are. Now, there's 2 colors. The receivers are the same, the purposes are different. The first thing we were interested in doing when we started setting up CATOS is, of course, studying lake trout spawning. We want to know where they're spawning, how well they're spawning, and something basic about spawning behavior, something beyond the application of trying to restore lake trout, but just looking at how do lake trout behave when they're spawning. Do the males come back to the same site year after year? Do they stay on one site, or do they go sort of shopping around to different, well, we call them bars, you might say, to different spawning areas, to different bars to see what's going on at different sites? What about the females? Do they come into one site, find a good male or a good group of males, do their spawning, and are they gone? Or do they shop around also? How long do they stay on a site relative to the males? This is just basic understanding of the biology and the behavior of the organism that we're intrigued to know about. It's surprising, after 60 years of trying to restore them in the Great Lakes, we still don't know some of this basic information. The other receivers on there, the yellow ones, sorry, I should say, excuse me, each of the yellow ones is on a lake trout spawning site that we know about. We've studied lake trout, I've studied lake trout now here for over 17 years, so I know at least some of the areas where their spawning sites, their spawning aggregations are concentrated. All of the other receivers, the red receivers are beginning towards a second study we're interested in, which is basically illustrated here. We have a fragmented lake. You guys know this. You know about the causeways. You cross Sandbar anytime you go to Grand Isle. This is the Island Line causeway. It's 3.5 miles long, 5 kilometers. That used to be open water. Now, it was a sandbar, just like Sandbar was. There were both rises in that, but there was open passage for fish across, particularly shallow water fish, or any fish during the winter time. But we've closed it to 2 very small openings, and in the case of the Sandbar Causeway, 1 very small opening. And of course, if you look at all of the causeways on the lake, we've got 10 of them. So the lake is now fragmented into the main lake, Mallett's Bay, Inland Sea, Missisquoi Bay, Carey Bay, the Gut. If a fish wants to go from east to west in the lake, they have to negotiate at least 2 causeways to get into or out of from the northeast arm relative to the main lake. So we're curious, of course, does that affect the behavior of the fish? Do they take longer migrations? Do they settle in different areas? Has that changed the size of their home ranges? Has it changed their spawning behavior? And therefore, has it also changed the genetics of the populations? Like Bill, we're studying whether these populations of fish, like walleye, like lake trout, have been subdivided into genetically different subpopulations that are no longer well connected with each other on either side of the lake or in the separate bays. So if we can track fish, we can put transmitters into these fish. At this point, we've tagged 70 lake trout, we've tagged half a dozen walleye. We can follow where they go from week to week, month to month, and year to year, and see whether these causeways are, as I say, changing. You know, do we see them bounce off a causeway, for example, and go elsewhere, where formerly they might have gone straight through from the main lake into Mallett's Bay, or Mallett's into the Inland Sea? Casos is available for any other kind of studies, and recently I've been contacted by the state because one of the species of interest, again, not from an angling standpoint, but from a conservation standpoint, is Lake Sturgeon. As you probably know, we used to commercially fish Lake Sturgeon, and we brought them to an all-time low. I mean, commercial fishing was banned, all fishing was banned by 67, but there are very few sturgeon left. They're a very slow species to mature and reproduce. So the question is, where are they going? Which rivers are they concentrating on? Do they go from river to river? Where are their wintering, their summering grounds? Where are their wintering grounds? So a sturgeon tagging project will start this year. To give you just a glimpse, because I really don't want to keep you too long, a glimpse of what these data do for you, I'm going to take you, our study, by the way, is very new, and so